We are very happy to have a fantastic speaker today, Somi Kordafshari from WeHi Institute. My name is Marta Gabrielska. I'm an account manager at Millennium Science. We are a distributor of 10x Genomics. And I also have today with me Jeff McDermott from 10x Genomics, Science and Technology Advisor, who knows everything about the technology. But we will be focusing today on Somi and her amazing hands. I think she was born holding a pipette already, because when she runs any of the single cell 10x assays, and she, I think, tried everything, it just seems to it's really no fail zone. So I'm really curious to learn more about how she does it, what's the secret sauce and secret ingredient there. So Somi, please, the audience is yours.
Well, thanks, Marta, for the kind introduction, and hi, everyone. I'd like to begin today by acknowledging the traditional custodians of the land on which we gather meet today and pay my respects to their elders, past and present. Well, as Marta mentioned, I've been running almost all 10x single cell assay in our lab, breast cancer lab here at WeHi, across human and mouse samples, different sample types, with over 300 samples to date using assays listed here. Well, I wish I had time to walk you through each of these assays and share my experience, because to me, every of these assays has its own unique strength and fascinating capabilities. But because of the time constraints, I will be focusing only on Flex, sharing some of my hands-on expertise with you, practical strategies, and my aim is to highlight approaches that ensure technical robustness, reproducibility, and consistently getting high quality data across 10x platforms, again, emphasizing on Flex. Well, a quick overview of my research focus in breast cancer. Breast cancer is the most commonly diagnosed cancer in women worldwide, with about 2.3 million new cases and 700,000 deaths in 2024. Here in Australia is the second most commonly diagnosed cancer, and a person has 1 in 15 risk of being diagnosed by this cancer by age of 85, which is about 1 in 600 in males and 1 in 8 for females. In 2024, it was estimated that about 20,000 women and 200 men were diagnosed with breast cancer here in Australia. Architecturally, breast comprises a complex epithelial ductal tree that ends to a cluster of acinis. I will show you to a cluster of acinis called TDLU, or terminal ductal lobular unit, which are the functional unit and basically where the majority of breast tumor arise. This epithelial tree consists of a bilayered two epithelial cellular lineage, luminal and myoepithelial, that embedded in a stroma matrix that has fibroblast, endothelial, immune cell, and adipocyte. And in fact, the crosstalk and signaling between these epithelial and stroma influences both normal physiology of the breast and carcinogenesis. Breast cancer is classified in a number of ways. Clinically, based on expression of estrogen, progesterone, and human epidermal growth factor into 3 broad groups of ER positive, HER2 positive, and triple negative. Histologically, into 2 broad groups of no special type and special, that I will walk you through in details in coming slides, and molecularly based on similarity of gene expression to the normal breast cell type, with the poorly differentiated Claudin law on one end of this spectrum that are remarkably similar to normal breast stem cell, and luminal A on the end of this spectrum, which are very similar to mature luminal cell. But my research centers on rare breast cancer subtypes, which are classified as a spatial histology, meaning they have a spatial pattern, a spatial histologic pattern in more than 90% of the tumor. They are highly heterogeneous, not between the subtype, but a single subtype like metaplastic that has both sarcomatose and carcinomatose component can show multiple histotypes. And the fact is, we've chosen these subtypes not only because they are rare, but also they have got the most aggressive clinical presentation, higher incidence of stage 3 and 4 at the time of diagnosis, higher propensity for lymphovascular, lymph node, and distant metastasis. And unfortunately, despite their rarity, they play a significant role in global breast cancer mortality, which is due to limited number of studies, lack of well-defined molecular target to define a systemic agent to do a definitive therapy. And a key barrier here is the scarcity of the fresh tissue, which has made it almost impossible to run a comprehensive molecular study to underpin the molecular basis behind this complexity. And that's exactly why we aim to run this project to overcome this barrier by using FFPE for the first time to look at the complexities of this tumor at high resolution single cell level, transcriptionally, and especially, as I said, by using FFPE, which we all know that they are the largest clinically annotated human samples. And what made that possible was thanks to 10x Flex assay in combination with SN Patho developed by Dr. Luciano Martelletto, which to me, honestly, this has been like a turning point, almost an explosion in what's possible, because for the first time, we as a cancer researcher can generate high resolution single cell data directly from archival tissue. And as I said, it's like a game changer, at least for me. And why FFPE matters? We all know that throughout the globe, the majority of human tumors are preserved in FFPE for diagnostic purposes, right? So which means that FFPE can represent a largest potential resource for identifying disease pathway, biomarkers, and even drug targets. However, a big limitation here is that because FFPE undergoes a lot of chemical and physical stress, this leads to RNA degradation, fragmentation. And in fact, before launching the Flex assay, almost all single cell assay by 10x, they relies on capturing the poly-A tail on 3' end, which means you would need intact RNA. However, in FFPE, as I said, it's degraded. There is no poly-A. So, and how Flex works, basically it's a prop-based chemistry. And it works based on in situ hybridization of prop to directly to mRNA. And because it's a prop-based chemistry, it's got the highest sensitivity across all 10x single cell assay. And you can capture, in fact, low expressed genes such as transcription factor. And in fact, in practice, it allows us to extract meaningful data even from degraded FFPE RNA. Just highlighting few benefits of using fixed cell or FFPE. Basically, by fixing the cell, you are locking lifelike state of the cell. You preserve fragile cells at the time of collection. And you can build up your own bank of samples, do batch process all at once, schedule your experiments, and run both longitudinal and retrospective studies. And on top of all, you can run large-scale multi-site studies from around the globe. And briefly about how Flex works on FFPE. Well, we extract nuclei, hybridize with prop, and then the beauty here is that you have the luxury of multiplexing either 4 sample or 16 per lane. Then the hybridized samples with probe will be combined with 10x barcoded gel bead to form gems, which allows us to profile transcriptome of thousands of individual cells at single cell resolution. And again, the most exciting part, at least for me, about Flex is the multiplexing power. Even with the newer version of Flex, which I've been using recently, the GemX Flex, you can capture up to 20,000 cells per sample. Meaning, if you do 16 sample multiplex in one run from one chip, you can basically get 2.5 million cells profiled. And here is how we applied this in our cohort. We have, these are the selected rare breast cancer subtypes from which we've got FFPE and their corresponding H&E, and then we applied these 3 complementary methods. And yeah, so you would appreciate that using this integrative strategy, we are breaking the fresh tissue barrier, while at the same time interrogating or profiling both transcriptional and spatial organization of this complex tumor at single cell level. Well, now let's look at the workflow. Actually, if I was gonna walk you through whole protocol, it would take probably the whole day or maybe a couple of days. But the important thing, please, to remember here is that the kit is already very robust. If you follow the protocol exactly, you will reliably achieve the targeted cell recovery. But the thing is, recovery is just one side of the story. And to me, the real key is the data quality, which depends on, again, if you ask me, I would say depends on being extra careful in a few critical steps. And please remember that each of your decisions impact downstream data. from nuclei isolation that I will walk you through a bit in detail in next slide, to the flex assay where you need to have gentle and proper handling, staining, and accurate nuclei counting, which are all essential, to library prep and sequencing submission that you need to consider how to choose index, how to pull, which kit to use, how many cycle, because these are all influence the distribution of reads in your final samples. And finally, very importantly, is that after sequencing, you have to carefully examine all the metrics in web summaries, including percentage of percentage of valid barcode, total cell detected, total gene, UMIs, median gene, median UMI, and sequencing saturation, and so forth. And because these assays are multiplex, you cannot fully control how many reads end up per sample, even if you precisely target cell recovery. And this can directly affect your final metrics and potentially the biological conclusion. So you need to be very careful on sequencing QC metrics before proceeding to data analysis. And yeah, so altogether, as I have shown in the as a title of this slide, the high quality data is earned at every step. Well, the nuclei prep, to me, it's the foundation of high quality data. But again, please remember, other technical decisions can also dramatically shape data quality. So some of the key consideration. One of the common questions that I've been asked is that how many scrolls you would need. While actually, I would say that while all the protocols mention mention certain number of scrolls. To me, the reality is that the yield depends on tumor area and cellular density, not just the rough size of the block. For example, you would see that all of these blocks here that I have put in this slide, they look like they have got a very big tumor area, right? But when you look at the H&E, you would see that, for example, this one, although the block was big, but it has only a small dense area. Or most of the time, in our case, working with breast, the majority, the highest proportion of the block could be the fatty tissue or fibrotic, which have literally very low nuclei number. So in a practical way, I would say that the takeaway here is that always consider tumor area, cell density, and cell type. Or for example, here you would see that between group B and group A, definitely if we want to aim for a certain number of nuclei, equal number of nuclei, the number of scrolls that I would need from group B would be more than group A because they have a smaller tumor area. And about the thickness of the scroll, I would always rather 25 micrometer than 50 micrometer. I mean, this is what I would do always, because I believe that thinner scrolls allow better reagent penetration, diffusion during nuclei isolation, and it would help you having higher yields. And yeah, as altogether, again, as I said, please consider the density of the area, how big it is, and the cell type. For example, if it is fatty tissue or fibrotic, definitely they wouldn't give you a lot of nuclei. Oops. And another few points about the nuclei isolation. I never treat all the blocks in the same way. So protocols give you a framework, but in a practice, every FFPE behaves differently. Some need longer deparaffinization, some shorter, some may need longer digestion, extra mincing. However, some of them may just come apart very quickly. So that's why I always emphasize observing each sample individually rather than following the exact same timing across the board. And for the digestion, I highly recommend a combination of the enzymatic and mechanical digestion. And I always use the RNase-free pestle, which I would assume that everyone would use that. And also please remember the goal here at this step is not just to fully dissociate everything. We don't want to overlyse the cells and end up with a lot of debris. The aim here, as it is mentioned in the protocol, is just to loosen up the tissue enough for the nuclei to be released. And I never go beyond 1 hour because, again, remember, from this step onward, there is going to be another digestion and mincing step, which would allow you to easily get nuclei. And one of those very little tweaks that I have optimized for myself over the time is that in the first step of filtration that you are using 70 micrometer filter, I would say be generous. If you still have a chunk of tissue, I would pass it through different filters, and then I would combine, and I can sometimes do a quick spin, and also then after all I would combine the flow through, because in this way you are avoiding the clogging and maximize the nuclei recovery. Or another tweak that I always use is that for the final filtration, which you would use 40 micrometer in nuclei isolation and 30 microliter prior to capture, I always dilute it in 1 ml, even if the protocol says 500 microliter or 200. No, I always dilute it in 1 ml, because again, I believe that if you, the more diluted, the smoother it would pass through the filter. So then I would spin down, and then I would concentrate it to the targeted concentration. And then because all the blocks we've been getting from hospital, literally they are colorful, every color you would assume that I have worked with. And at the beginning I had this concern, what if this surgical dye would interfere with Felix assay? I can confirm for you that I have used a wide ranges of color, and none of them has had any impact. So don't worry about the surgical dye. And then for the counting, I would highly recommend using fluorescent dye rather than trypan blue, because FFPE derived nuclei are very debris prone, and trypan blue can basically stain the debris, meaning you would overestimate your cell count and underestimate the debris. So I always use fluorescent dye, specifically PI, which is an intercalating dye. It means specifically binds to DNA. It means you have lower chance of counting debris. And you know that both trypan blue and PI, they would penetrate and diffuse into a compromised membrane, but the difference here is that trypan blue stains the cytoplasmic protein, while the PI, as I said, specifically stains the DNA. And also, prior to counting with automated counters, I always check the nuclei quality under the microscope, either with dye or without, and then I would use the automated cell counters. And here are some examples of pre-hybridization, post-hype, and pre-capture counting. Shifting gears, I'm going to show you some examples highlighting just how critical sequencing QC metrics really is. In fact, they are not just a reflection of the technical performance, they directly shape the data we see, and more importantly, influence the biological conclusion that we can draw. Here in this graph, I'm showing you the 2 sequencing run per sample, green and pink, the total reads from each sequencing, right? So now you would see how significantly they impact these sequencing, the depths of sequencing impact on the total UMI, which is the most critical part as defines the complexities of your sample, which would give you a truer picture of the underlying biology. And again, here, this graph shows you how deeper sequencing increases mean reads per cell, and more importantly, excuse me, and more importantly, how significantly improve the saturation. And here within a single subtype, you can see a strong correlation coefficient between the sequencing depths and total UMI, and also the median, excuse me, and also the strong correlation coefficient between the mean reads and median UMI, mean reads and median G. And more importantly, again, you would see that I have shown that, for example, in this particular subtype, something around 50,000 reads per cell, at 50,000 reads per cell, the saturation starts to plateau. And I'm going to show you how these metrics impact meaningfully to cell type and clustering. So you would see that in a previous run with a lower sequencing depths, there is no T cell and B cell. But look at here, we've got a big B cell and T cell cluster. Or previously, as you can see, the clusters were merged. There are some bridges among the clusters, and now you would see a clear, distinct separation across all clusters. And this is alongside the all the cell type proportion has also increased. But the more importantly, as I said, is that we are now getting B cell, T cell, and very nice and distinct separation of the cluster. Another example, again, previously we didn't have plasma. With a deeper sequencing, now we've got a big plasma. Or look at here, T and B are literally merged, but now here we've got T cell here, and very far from there, it's the B cell. And also with the deeper sequencing, we've been able to see a distinct neutrophil clusters. You know that neutrophils are hard to be captured. They have way lower transcripts, but as you can see, the deeper sequencing has enabled us to see these cells. And another example, again, there is no B cell here, but we've got a big B cluster in the deeper sequencing. And here T cell are literally merged to tumor cell, but look, T cell are here and here is the tumor. So we have a clear separation of T from tumor cells, which previously, as I said, were merged. And here I'm just showing an example of one of our subtypes that have 11 patients, that you can appreciate that across all patients, how well resolved and consistent are the clusters. And we've got a full spectrum of all cell types in all 11 patients. Another subtype that has 15 patients. And again, we've got a full spectrum of all cell types consistent across all patients. And here I'm showing you that this is an integrative analysis of all cell types from the previous two subtypes I showed you at individual level. So here I show you that how integration reveal reproducible and cellular landscape across these two subtypes, and how the integration maintains distinct clusters meaningfully. The data quality, and it shows that how the data quality and integration has worked very well. And we can recover and compare the complete cellular composition for the subtype. So with this, I'm gonna wrap up things for today, because today's webinar meant to focus mainly on hands-on experience. So, but I'm going to give you a summary that yes, we've done a large scale, high dimensional systemic analysis on rare breast cancer subtypes for the first time using FLEX on 80 samples. And alongside that, and maybe in future, I will show you my result. Alongside this FLEX, we've done a comprehensive spatial analysis using Xenium on about 50 samples. And we've got high resolution data. We've been able to show cellular dynamics, unique niches, and cellular interaction. And I really want to quickly make a note on Xenium that honestly, for me, the resolution is breathtaking. It's unbelievable that how clearly you can see individual cellular interaction across the whole tumor, how cells are mingling. And I will show you in future, you will see that when you look at this cellular interaction, it's like a crowded cocktail party or a dynamic dance floor that cells are connecting with multiple partners. Anyway, that's it before another time, and that I'm really looking forward to share my Xenium data to you all. Yes, and yes. I also like to highlight that, you know, that I'm a wet lab scientist, and I want to highlight that all the large scale data analysis in this big project has been led by our highly experienced bioinformatician, Andy, together with his former PhD student, Jinming. And also I want to highlight that Kuei and Kat from 10x, they have been incredibly supportive and helpful throughout the entire project. I really appreciate all of these guys. And I'm gonna thank all the people helped me this project possible. But first and foremost, I wanna appreciate my two amazing supervisors, Jane and Jeff. Honestly, Jane is incredible, always inspiring, full of ideas, super supportive, and very kind in her approaches. And I would say that without the way she's been backing this project, none of this would be possible. And maybe you wouldn't believe, but over the past 4 years that I have this privilege to work with Jane, every day for me has been like, as exciting, as motivating as the very first day. And every day I've got more enthusiasm and passion to dive deeper and deeper into breast cancer and learning. And also, I've been very lucky to be surrounded by amazing people within our lab and beyond. And yeah, with that, I will leave it with you, Martha and Jeff. Thank you, everyone.
Thank you, Somi. That was so inspiring. I think you are the first researcher that I've heard was talking about, actually, we were talking here about flags, but when you were talking about xenium in such poetic way to describe cancer, that like it's just unbelievable. It's just Somi.
I really wish that I could show you, but I
I
knew that
know.
I wouldn't have time. But
Yeah.
definitely, yeah, I will do it at next
Yeah,
year.
we will definitely. We also have, for everyone in the audience, we will also have spatial conversations. So when Somi is ready to talk about her xenium experience, we will invite her to the spatial part of this webinar. Just a reminder to everyone, you can either raise your hand, put questions in the chat, or in the question and answers, and we will read them aloud. But I just wanted to, while everyone is still a bit warming up for the discussion, I really like how you were, every time you were referring that the sample prep really is reflected in the shapes, the data quality, because this is what actually you want from assays. You need the
Yeah.
results to analyze. So that I really like that. So a question from me, hence we are waiting. So in my life, when I talk to researchers, sometimes we are encountering, that FFPE blocks are coming from different places, right? Like
Mhm.
some surgeons can put it sitting on the sample out of the patient for a while. Did you do like a screening? Do you have the information for your samples? Like how long were they sitting? How long were they being fixed?
Yes, that's a really good question, actually. It is really important, Martha, how these FFP has been made, how long they have been sitting in OR, on ice, room temperature, how long they have been in the formaldehyde. Um, I have to say, regarding all the 80 blocks that I have processed, I have been lucky because they all came from RMH, as you can see, the Peter Mac. And as far as I know, they are following a fixed protocol. They wouldn't go beyond 18 hours of fixation in formaldehyde, which I would assume that it would really impact the RNA quality. And I have to say that ideally you need to do DV200, but I have never done that because, again, I would say that if you do DV200, you see the quality is good, but again, how would you know that after nuclei isolation, the condition and situation is the same of the section that you took for the DV200? So I haven't done that, but sorry, your question was that about the fixation. Yes, it is really important. And as far as I know, it shouldn't go beyond 18 hours being in formaldehyde.
Yeah.
Yeah,
And
I'm
like
just,
I have Jeff, our expert here, so I guess he doesn't say no, you're wrong. So I guess you're right. Yeah, so that's really important. And I know some people are struggling to get either the information or have sort of agreement to get the samples processed in a certain way. Also another question from me.
yeah.
So how long, so when I joined Millennium like 3 years ago, I think it was around that time that Somi was sort of getting started with single cell. And how much practice did it take for you to become so confident when you run the essays? I think there might be many people in the audience who are just about to start the journey. It might be a bit, scary. So how did it happen for you?
Um, well, it's very simple. The answer is that I would really recommend just reading, diving into the details, know the rationale behind every reagent, every step. Don't rush in to do the stuff in the lab. Just make sure that you have a comprehensive and thorough understandings of every step, every reagent. Honestly, this is how I've been doing this. I wouldn't touch anything in the lab unless I know the full information about the reagents, everything. I
Mhm.
think this is the key.
Okay, yeah. And always feel free to reach out to your friendly 10x team who will answer all the questions.
Oh, of course. Sorry. Yes, of course. Yes. I mean,
Yeah.
Jeff and Quay, everyone in 10X, James, they are incredibly supportive. I mean, yesterday while I was walking the street, I mean, Kat is super nice and supportive. And for this flex specifically, she's been very helpful. Whenever I have another key that I have to mention to the audience is that never do anything if you have even a small amount of doubt. I always call Kat and I make sure that I'm doing things right if I have doubt. That is another thing. Yes.
Yeah.
And we are very lucky to have her, Jeff, James, Quay, yes.
Yeah, all right. But I absolutely agree with you. I think our audience is very shy today. So if Jeff doesn't have any more questions, I think thank you so much, Somi, for talking about your data. It was really fascinating. And like, I still think there is a bit of magic in what you're doing with all these tiny decisions you need to make, but that's okay. We will meet Somi again. There will be another cellular conversations about different assay. It will be multi from memory, I think. So please stay tuned and monitor your inboxes. So thank you again. Thank you, Jeff, and thank you, Somi. Thank you, everyone in the audience. We will see you next time.
Thank you, Martha. Thank you, Jeff.
Thanks,
Thanks,
Somi.
everyone.
See you,
Bye.
bye.
Bye.